Good morning to all. Put your things in your attendance. Yeah, good morning again, once again. So now, is it sir? Yeah, shit. It's okay, but I think it will be otherwise not going to connect. It will require to load, I think. Many times I forget many things. There is my error. Fine, I will be here, so it will be better for everyone able to listen. So today, on the next topic, we are looking for 2.1 methods for isolating pure culture. Methods of isolating pure culture. So what is that? So first we are looking for few basic things. Culture. Which define as a one. I have already covered this. What is the culture? There is a microbes grown in a medium, right? So bacterial culture, or the sometimes it is been known as a fungal culture. So there we can say the fungi is growing in a medium, bacteria is growing in a medium. Then we are going to call as a bacterial culture. Sometimes it's considered as a microbial culture. So what is being asked, you have to write, right? Thus, whether it is being asked as you are Indian or you are a human, accordingly we need to write. Or you may write as a I'm from the indigenous from so and so place. Okay. So it is been asked as a culture, microbial culture, bacterial culture, fungal, or anything else could be. What is been asked, accordingly you need to write. Then the mixed culture, it having the more than one microbes. Mixed culture, that means how we can get the generally mixed culture, either it is from the soil sample or the patient's stool or the blood. When you're taking the blood, we don't know in exact that what sort of the microbes could be there. So in it, the pure culture, that means is having the only one strain, only one kind of a bacteria are present. If it is like E. coli, all are in a E. coli form only. Okay, so all the bacteria are of the same kind of a things. Okay, strain will be there. If I'm going to ask here, then all are the human, and otherwise you can say the Homo sapiens. No, anything else is here. If sometimes having some pet dogs with them, in some universities allowed, but they have to kept near to the their pet house are there. In foreign countries they allowed. If you're going in somewhere, there if you're telling that I have my pet, I cannot ignore it. So you can take it. Otherwise, can say some other childs are there. Some are having the child, even the students of the faculty. So Christian houses are there. So over there, they can have their child to be get given. Therefore, they're taking care. Some members are there. So you have to pay additional. So that can be possible. Okay. So it is like that only. And the exenic culture, a culture in which all the organisms are of the same strain. You can see. Can you see the difference between with both? Pure culture and exenic culture. Is there any difference between it? Culture in which only one strain, but culture in which all the organisms are of the same strain. So generally, we are doing the terminology of pure culture. Okay, but the other terminology is the exenic culture. Both are the same. Yeah. Uh, genus, then the species system, right? Then the third terminology we are calling as a strain. That means isolate. If you are isolating E. coli, maybe from the several places from the Gujarat. Okay. So what's going to happen? Minor variations could be there. It is like we are having the some a caste. Okay. Some of the castes are there. All are the human beings. There we are going to call as a race. For a human population, we are having the some terminology is a race. So some basic are changed. So in both, here we are looking for what? There is string or all organisms are the same string. That means I have isolated equal. So that from the maybe you have taken the water from the Marwad hostels. One is taken from the your home. So if you are going to check at their levels, strain level, okay, we going to say them they both are equal ion. Okay, but several characters are going to change into words. So that is at the isolate level, some changes are there. So we need to talk about the strain itself. So generally, we are going to not focus more on it. We are going to say like all the organisms are of the same characters. Sometimes it is called like that. Okay, so it is the another terminology generally being not used into all the literatures. Exenic, this terminology is a very old classical microbiology. Over that time, people were using. So that is why I just introduced pure culture. That means no any other organisms are there. It is only single. But this exenic terminology, it is from the classical microbiology. Just I am looking to introduce. Then the colony. It's a cluster of microbes growing on solid medium. Colony only can be able to observe on a solid medium, not in liquid. In liquid growth, it is floating. So there we are going to call as a culture. Okay, that is a culture. Generally, culture can be called for the solid and liquid forms. Same terminology can go. There we are not defining. There we are just defining what in first that are grown on a medium. Whether it's a solid or liquid, we are not going to differentiate. Okay, but when the term colony is coming, that means it's required support to sustain and develop. So all cluster of the microbes growing on solid medium. Okay, it can be of any solid medium, that is a nutritive agar or any other agar or the gelatinous phase. Solidifying agent is being added in a medium. Then the growth of microbes are happening, which we are going to call as a colony. So in nature, this is the why we need a pure culture, and importance of a pure culture, or why we are looking to go for it, essential or the importance. In nature, what is main part? Why need to have a pure culture? In any experimentation or any studies, when we are going to do some experimental part, whether it is related to pharmaceutical or medical sector or agriculture, or two categories classifying growth. What is the beneficial microbe and another is the harmful. Like you are having some friends, like you know there are two categories are always taking care. One is always taking your advantage. One is always giving advantage to you. And another is no. It is like a okay, having the coin, zero. So, but classify. Some are the benefit you are benefiting to you, some are harming to you. And another is no any effects. It's like that. 10 students are sitting here and 10 students are sitting here. Just coming here, there is no any kind of interactions. Though interaction is there, but they are not going to benefit one to another, not going to harm. Now the another friends group is coming into it. That's one having very much advantages for the another. Maybe it's a group or just one or two friends. Another it is always harming. Maybe it is of a so and so reason. So when you're looking to study any microbes that are beneficial or the harmful to whom? To whom? Yes, yes, give the answer. Human. Okay. To whom? Sometimes I'm going to ask the same questions. It means there are the multiple answers. Okay. Human, I understood. Beneficial and harmful to whom? Plants. To whom? Okay. All the yes, that is a form of human, plant or animals, or the environment. At the last, okay, at the last, we can consider the environment. Some are benefiting or the degrading something or making a harm, but it is according to our view. Okay, this is according to our view, they are benefiting or harming. Otherwise, they are sustaining themselves in a food chain. When you are looking in the environment, it is a sustainable for them. And it is always like that one species try to overcome onto another. From since long it is coming. Okay, it is like that if you are going to control one or in a food chain, if you are taking any one organism, the whole in a system or in a food web, some is going to get hampered into another, one to another. Right. So when you are looking to study, either it is in an environmental level, at a larger in food web, or you are looking to understand any microbes. At the microbial level, we need to have them isolated. Okay, in a pure form or in a single form, which is most essential if I want to. Generally, I'm coming in class. Okay, but when I'm looking to, like in a, what is your level of intellectuality? Common answer is always coming. But in during the Viva, we are isolating you one by one. Okay, we have delivered. Now your time to revert back. In a class, you always telling, yes, sir, everything understood, 100%. More than that. Right, when you are coming in Viva, so that was, yeah, I think, and the other parts will start to come up. Okay, so we are isolating one by one. It is like a pure culture. Why? We need to study over that time. That what was the gain you have done in a class. Okay, so we need a pure culture. So when you are looking for beneficial or harmful, like a yogurt is there. Okay, mixture of the microbes is there. It is beneficial to me. So I will populate them in a one culture in a medium, and I'm going to use as a my nourishment. Sometimes in many medicines also, they are the only a form is given, powder form is given to the people, those who are during sick condition is there. Digestive system, metabolically, they are not much active. So we need to give supplemented from the outer side. So during that time, they are going to get enriched. Their body is going to get the normal flora back. When you are looking to do a study on the harmful organism, what could be the things? In harmful organism, the main thing is what? That we need to isolate. That means they are pathogenic. I want to control them. So what sort of treatment should be given? It is like the some guys in very much notorious in a class. Yeah, initially we need to give the some warning. Second day, level will increase. Third day, I need to increase and then collaborate with the superior to me. Inform to them. They are going to call them in a chamber. If it is not get treated well, it has been called to their parents or the further stages is going on, we need to call the police station. It depends upon the level. So what is the pathogens over there or the notorious thing that accordingly we need to treat. So we need to first isolate to treat them. That is first to second stage. When the pathogens are there, I want to control. So what I am going to do? Antibiotic sensitivity test. Right? These microbes either is maybe following the gram negative or the positive. So I can go for the broad spectrum antibiotics first. Sometimes broad spectrum antibiotic is not going to work. I need to go for specialized or I need to understand their basic physiology. What sort of cytoplasmic interactions are there with one to another in pathogens? Accordingly, I will try to give the antibiotics. Is it like that? No, always it's not. Why the doctors are prescribing so many things? If we are having the only two kind of organism, gram negative and positive. But even after, there is enormous number of the medicines are available. The reason is what? Each microbes needed different treatments. It is like that. I caught the one notorious guy. Okay, it may have the so much power, influence. And if I want to treat, that means it can change or modify the place or the decision is being taken. So I need to go further one. So one antibiotic I have prescribed, it didn't work. I will go for the second. Then the microbes. That means the notorious guy is able to manage it. I need to go for the third stage, fourth stage. So I have then after characterized, after so many years of experience, if I am able to filter all the experience, that if this person is there, I need to treat, I directly need to go for the fourth stage. No need to go for the medicine 1, 2, and 3. It's a wastage of time. Like, so control the microbes, I need to give the prescription number 4. Same it is there. If I want to control this guy based on the character, I come to know that if I'm going to in the first stage, second and third stage, he has so much influence from the outer world that my decisions or the decisions taken by the one authority is going to get changed. Right. So all the alternatives and are there, we need to understand. So when you are isolating, what is our purpose? To do a treatment. When the beneficial microbes are there, what is our purpose? To get some gain out of it. Okay, some benefits we are going to look. It is also there. That was till the notorious. Now I'm looking some research-based work. I got the research projects, and I needed some students who can do some good work in my lab. So I am going to check it out. Who is punctual in the class? My first criteria. If suppose who is good in handling. Okay, then the certain criteria I have set, and within the 1 or 2 week or a month, I'm able to identify this person is worth to take in my lab. So I can give the benefits that experimentally, then the practically they can enhance their, you can say the capacity. Okay, then I may give them some certifications for the techniques if it is so, or I can give the further recommendations whenever they are looking ahead. Because I then after come to know at the personal level, what is their intellectual capacity, right? Sometimes may happen that I may be here after a few years, I may not hear. Okay, I was in previous institutions, but when you are going in somewhere in some interviews and others, right? Based on your previous profile, they come to know, okay, sometimes my many friends are there in several farmers. They calls. Do you know so and so names has came for the interview and we are on a final stage to take them. Okay, so I know that yes, I was in previous institute. So many guys I'm knowing, and this if you are taking, that will be a really good for your institutions. I can give the some suggestions or feedback, and another way also. That is up to you. I can just give a hint. That is up to you. I'm not aware much. Just one statement can take you at the one to another level. Right. So I can identify notorious guys. I can identify very prominent, very prudent student even. And other are in middle. They are coming, no any harm, no any benefits. They are learning, they enjoying their life because it's a college. Okay. So I can give the feedback like that. So what was there? My motto, not to classify you people, but for a Viva and other things, I'm going to check it out, look it out, your level. Right. Accordingly, I'm going to give you marks. That is the main or duty of mine. That is no any benefits or no harm. If I'm going to say everyone is good or everyone is bad, then the judgment criteria for me, it's going to get ruined. That this fellow is always telling that everyone is good, everyone is bad. There is no judgmental criteria. Then the one person is may ask, then after they are not going to ask again. If there is if the person is not having the judgmental eye into it, judgment is not there, there is no any kind of a gain to ask him. So such thing is there. When you are looking for beneficial or harmful, we need to get in your phone. Okay, your phone is there, you can play with them. Okay, so how do we going to get such? There are the three techniques. Each technique you are going to learn. Part, okay, every techniques are mentioned. Plate techniques, spread plate technique, and pour plate techniques. So each are based on, currently we are talking that is solidifying medium we are going to use. That means it's related to plates. That means it is having the solidifying material. So what are the basic things? Stick plate, spread, and pour. Have you seen such kind of a things in your time, or anybody has called? What is it? Blue. Wire. What is it? Nichrome. Wire. Nichrome wire. What is it? Yes. Okay. But there are two images are there. Above one is also found. Have you seen it? Anybody? So let me just increase its size. Then it may be going to see. Yeah, can you see this what's image? So in general, it is been practiced as a microbiologist. That is a below one, right? That is you are telling is the nichrome wire loop. Why we are using the loop to take the some suspension. Okay, bacterial suspension or a microbial suspension out of a tube or a flask to transfer to somewhere. Where we are going to transfer? Either it is maybe a plate or a broth or any kind of a, you can say, slide even. If you are going to do any staining, you are taking the full culture and you are spreading. Sometimes that is you are transferring from the culture. It may be from the liquid as well as from the solid as well. Like bacterial colony is there, you are having one plate. You are going to just touch it on the bacterial plate colony is touched, and you are transferring either on a plate again, or you will be transferring to cloth. In any medium, you are maybe transferring, or solid surface there you are spreading over it. So we are using such kind of a things. Here we are using the nichrome, not any other metals. What is the reasoning behind it? We can use the iron. So nichrome is, we can go for the other metals too. But the basic part is what it should get at a given time period, right? It should be get the for the sterilization purpose when you are looking. It should be get a higher temperature. It should get acquired within a fraction of time period. It should get cooled down. So people have gone for the several aspects, and from there they have found this. If you are going to, this also right, melting point is very high. They are going to get acquire the temperature, but the melting is at a very higher level. It's not going to get melt if you are having the iron or the copper. And another is what, they are not that much reactive. That part you have to add, right? If we can say the gold even, silver to gold, and the platinum, why it is much and much and much costlier? It is non-reactive. Non-reactive metals are having their higher values. Why? It is because when you looking to any metals to be wear on your hands or on any body parts, what is going to happen if it is like a any copper or any other else is there? It start to oxidize on your surface. There may be a reaction is taking place, and your skin may have the irritations at the later stage. So metal should be of a non-reactivity. So here in microbial part, we are looking to use it. Another, it is a wire loop itself, but made up of just of fibers or plastic. It is been already been sterilized by using the gamma radiations or others. Okay, how to use it? It is totally packed coming. It is this wire loop, you know that we are going to flame it. This is totally closed. Individual, this are coming into closed packed plastic wrappers. It is like a you are using the wafers. It can be a crunchy and munchy for a month or a given time period, right? Is it so? If you have taken the like one potato wafers for the lace or the balaji, okay, you have kept in your bag, right? For a 1 month, you just forget to eat even, but after 1 month, it is going to remain as such. But if you have opened it, then after it start to stale you, the fruit quality is going to reduce down. Similar to it, like syringe, if you are knowing, syringes are coming, they are closed. Okay, injections of syringes are coming, they are sterilized and remain closed. Similar to it, the plastics are coming, but they are in routine practice. It is not being suggested to use because the cost is going to high. Okay, if it is just of a 10 or 20 rupees is there, you need to do every day a 50 to 100 works. So it will be more costlier. But sometimes it is prescribed. The people are using. So you need to understand and learn that not only a wire loop, okay, that is we are looking that loop structure, it is of metal. Other are also available. Okay, now the several points are coming. That you are going to take either liquid or solid, and you are going to other two terminals are coming. One will have to when it is splitting and another one is spreading. So you can get the document and same by our practical session. Okay, one is a streaking, another is a spreading. When you are having the loop with you, it is going to be always a streaking. Okay, spreading is going to come when you are going to use some glass rod. U shape or the triangle shape glass rod we are going to use. So I am going to show some documentary. Based on documentary, we are going to understand well. So quadrant method, P-Sc, continuous sticking, radiant sticking. That means what? I do have one plate with me. So what I'm going to do? First, I should have either plate or the liquid form. It can be possible at a larger level. And take one hand and work right. What I start to do? First, you will requirement is what? I need to tell you small wire. Okay. So I must come with that once again for it that you are not able to manage your culture by one. Okay. So now we need this master. Here given number, you are going, they are giving then you to read the stronger. Something is that one. So you are senior. You are not into the experimentation. Okay. So what that my loop should be there, it should be one part what bottom part that is half of the medium step. And here this is what I'm doing. So okay. Right? Five fingers. God has given to us. So that I can use. So I will close by closing distance. So there is only that one. There is just minutes. What are the things? Then another thing. Those are having long heads. Others have some limits and other different colors. Those who are in the extra process, it should not get out to the hair. Otherwise, your hair or other plants. And it's one safety that you have to manage. Okay, otherwise, it's going to interview. And of course, I need to find how to do that. From the back. Now, in this slide, over plates, I am going to describe of the strict plate now. The strict part, what I mean that on surface. I tell you that not to form. Okay. That's that and lightning you are going to split. Speaking very simply that similarly normally use it to under place. Why the reason is that let is there in your hand, you are going to get split over the surface. Now, most important part who is putting sticky. When I was started to do the sticky up, near my then 5 or 6 times you are stick or 15 times you are stick or 25 times you are stick. Okay, that is not essential to guarantee your health. Why? Whenever we can look to culture in my hand from subsequent time, they have a limited number of bacteria. Okay, when you started to stick, not spread. When they started to stick, it started to on the nitrates. Okay, this from the look when you are doing that is one by one, maybe with the initial part, thousands of microbes were there. And moving forward and forward, okay, it's like a fuel is going to reduce down. Then the number of bacteria are going to reduce down. So when I'm speaking at the end, after 20 to 25 seconds, at the end, and just have the isolated colony. If you are going for 5 times, done. Right? Again, say, start to understand, start to learn, start to learn. On a second, now another thing is that you want to contamination. That's the most important essential part. Because that open speaking. That very open, while open. It is going to be someone else. Okay. By chance, mistake, sometimes it's something. Okay, the idea was there. What was it down? Let's okay, but by chance if I come in, then it's not I'm not looking to harm someone, but it was mistaken anywhere, whether it's a glass, any material, because many users are not doing glass, going to cover the surface or sometimes what happens is the glass is broken down to minor parts that here during the habit to sometimes okay or many things is different. Next day, next time, so right, so there is going where the that's the sensitization or that is good. But if you see the further, then you are trying to remove every item or the and immediate portions in a particular nothing is going a problem. But by chance happens, if you are yes, you are telling yes, okay. Otherwise, I don't know. Nobody knows. There is not a problem. But the other harm going to harm is there and it's very common. A good person, now we are starting to see how do we close keeping the methods are there into stacking are this. These are the certain wire loops. Can you see? Wire loop of a different design. First one is having some loop. Second is open. Third one is of L shape. And last one is having the some. Transport there we are using second one. Just to then yes. But I will do I touch the part by using the my second loop to my video. Just that and the very less number of yes. Forward one is there. Okay. Sometimes we use two. We are making that two is thing. And using it part. Okay. Fourth one is used when you are doing something. can use here and there. It's not very common when we use these or that. But depending upon the experimental design, we may go for one to another kind of a wire loops. These are in metal, same are available in plastic also. But people are, if there are luxurious places there, you may have it. Otherwise, we may use this kind of a. Now the quadrant. Quadrant name is itself telling. So what I am going to do, like ABC. First you can see this triangle. Yes, it seems. I have taken the plates. What I have done, honestly, all around. What I am going to do, this one is last thing, I think is yeah, stick plate, continuous stick. Okay, you start it. Okay. Continuous stick plate method. Here you can see at the bottom the number of the bacteria is less compared to the upper. Right. So it is the difference over here. We are interested to explorated column. Pure culture is our main. So we will go for another is called the Spartan. This kind of a things you need to learn, and this question can be asked in the 4 or 6 or 8 marks. So just I'm showing the difference, right? But you need to explain step by step. You have done this way, so you need to put it down in like that. Okay. So what is the first thing? That transfer. I have taken the material. I have made it. Now I am not going to directly what I do, I will select where is there across. Right? This means I will start. So where I will start, actually the important part is what? The people having the some different base to do. So what they will start to do from there. The looping part from here is going to touch another part. So if it is continuous, then the isolated column may not. I have started to rub my loop on the surfaces. Again, I will close it. I will rotate. That is why I call to rotate your plate. Okay, rotation of the plate is required. What you can do, you can do it also. Sometimes, if the rotation is done more, then also a problem. Okay, so what you need to keep in your mind, you can draw it even. You can draw it even, otherwise you can manage. So, short sticking. I will stabilize my loop. Don't forget to stabilize. So when you are looking to get a pure acidic colony, there are several variants and several methods also. It is choice of the experimental part. Is looking that I will not go for this kind of method. Somebody is going to say this another method. It is a choice of the research. But for learning, that is the number of microbes on a loop. Almost there. When I started to do continuous scaling, number of microbes are reducing. Okay, but what I will have stopped at 13 level. Here you can see actual plants. So here, third, so I'm looking for the getting to some isolated colony. So I may go for this is called the 4-time I have used. Okay. Okay. So it's called the Part-1 method. Then the another is a T-stick. Another is a T step. So what is being done? First step over here, sticking is done. Like the second. What the other people are doing? That they are not going to rub. In the first leg of the what I have done, I have made a line. Second line, I have closed. Exam first, when completed, I have sterilized my loop and I will touch this last time. Take the last time. Or in a T. First, this is being done, then I will go in the second section. How does when I will say? Already it is being drawn. One of plates. Okay, some plates may be here. I can show. Yeah, here it is. What is there? They have done the first. Then you are going to touch at the end, where you have ended. Just touching your loop, and then you will start to spread again. Maybe people are having a changing their areas. Okay, so it is always better if you rotate. Suppose I will start to do in this way. Rather, you may rotate in a way that you may spread in this pattern. So you will get the larger area to strict. As you are going to reach at the end, you will stop. But it should and must not cross in a front. Third is there, you must sterilize it, you just touch, and you may start. Not from here. Yes, always. The eighth stage, we need to sterilize it. It's a very important thing. Sterilize is not like anyway. That's what I told. That depend upon the person who practice. Another. Next, it is stick plate and the radiant stick method. What is been done? Radiant stick method. What is been done over there? Either on one direction. Yes, it is not continuous. Just look, I'm going to upper side. I'm not going to stand like for a while. Just inside make the lines. The next slide you start to make some lines. Then you have that. That some colonies, those who are sticking here and there, are going to evenly stick on the other place. So here you can see it's called the radiant. This method is called. That is the distribution of the distance. You can have the T in any format. So, or you can have the substance, or you can have the various form of activities. That is the word is just Yes, I will show the or advantage in these students. I will discuss maybe later. Okay, okay, so you put your attendance in the given link. You are not notified. I am not so sure. Maybe sometimes because yeah, it's okay. Fine. So till this time ending here. In the next lecture, we'll move ahead. So till this, anybody having any doubt?